So today, Matt, you're going to be working on the tin checks and the time-constrained searches looking for the vertebrates and the amphibians. We've done some of the sites, but we've got some yet to go, and we'll run through which sites to do. And the two of you, Zephyr and Jessica, we're going to be working together on doing some pulling of canopy samples, trying to bring down the canopy samples and then measure them for their water stress levels. Are there any questions?
No, I don't think so.
All right, awesome. Matt, you're headed that way. The rest of us are headed this way. We're at Tampico Ridge, which is about 20 miles south of Western Oregon University, and it's in the heart of the Oak Cascadia ecosystem, which stretches along the west coast of North America from Northern California to British Columbia.
So what I'm doing right now is I'm tying a noose to this lead weight that we're gonna launch up into the canopy. 3, 2, 1. Got it.
So the research we're doing here is trying to understand the ecosystem and all of the endemic and important species and biodiversity that lives here in this ecosystem. So this land was historically the home of the Kalapuya and Lucky peoples who cared for this ecosystem. Over the last 100 years, this ecosystem has declined by over 90%. We now have 5 to 10% of this oak habitat left. And all of the endemic biodiversity is endangered from the loss of this habitat.
Samples.
So there's a lot of different types of samples that we collect out here, ranging from trees to animals.
I'm gonna start by doing the sweep net. So I like being a part of this research because it's hands-on and I do enjoy being outdoors a lot. So the next step is to fish out the ones that are big enough to input in a jar. I typically like to use my hands, but you can use tweezers. And these are already processed samples from the lab, and they are individual invertebrates put into their designated families. As a pre-med student, I am very excited to learn more about the invertebrates, mainly because there are a lot of tie-ins.
Hey guys, how's the collecting going?
Pretty good.
You guys get them all?
I think so.
All right, I'm ready for them.
All right. Here's the first sample.
Where's this one from? Woodland Road 10L. And we're gonna make a small cut of one of the leaves right at the node of the bud here. Then we're gonna put it into the pressure chamber. Okay, so now we're going to increase the pressure in the chamber, or to see when water will come out of the end of the cut petiole. 37 bar before the water started coming out of the petiole. This research has taught me how to be a wildlife biologist and how to speak publicly with other biologists about the work I've been doing. I know it's definitely not for everybody, being able to be out in the wilderness, hear the birds chirp, hear the trees crack, everything. I like being out in the wilderness. If it's for you, you'll love it out here.
I am just so excited to be out here studying the ecosystem, immersing myself in it with our Western students, and helping them fall in love with both the ecosystem they live in and the process of doing science.